So we've got Genomics Lite talk about parasitic worms in focus, which Sarah will be discussing with us. This will be followed by a Q&A, and we'll try and wrap up by 5:30 p.m. So the session is being recorded and will be available for you on YouTube and on our website. And that is all from me. I will make just one more poll live. If you guys would mind this one out, it's just for our records. And I will pass on to Sarah. So Sarah.
Great, thank you. Okay. Okay, is that right, Cindy? Is that good?
Yeah, that's all great.
Okay. Hi, my name is Sarah Buddenborg. I am a postdoctoral fellow at the Sanger Institute, and I study parasitic worms. So you might be surprised to hear that greater than 50% of all organisms on the earth are parasites. So a parasitic lifestyle is the most prominent of all of the lifestyles. And organisms have evolved into parasites from free-living ancestors at least 223 times. So it's really advantageous to live as a parasite. So in the chat box, I want you to think about what one word comes to mind when I say the word parasite. What do you think of? Anything? And then Cindy's going to share some of the answers, I think.
Yeah, so we've got one so far. Is that 50% by number or species? Someone was asked.
So 50% of all known species, or greater than 50% of all known species, are parasites.
And we've got a few responses for what do we think about when we say parasite. So we've got worms, leech, deadly, thief, invasive, gross, insidious, infection, harmful. Some really good words here.
Yeah, those are very descriptive.
Very.
That's awesome. Good. Well, it seems like you guys are grossed out by parasites, but I hope to tell you how cool they can be, and you can see how exciting they are to study and maybe have a bit more appreciation for them by the end. So what exactly is a parasite anyway? So we might not know yet what is considered a parasite and what the definition is. So a parasite is an organism that lives in or on another organism of a different species and causes harm. So we can think of this as a type of symbiosis that is positive for one organism and negative for the other. So the parasite gets a benefit, and then the host, which has the parasite in or on it, has a negative effect. And you've probably heard of symbioses, but they're a way of describing how we live in relation to other organisms around us. And there are two other kinds of symbioses. Does anyone know what these are called? You can answer in the chat if you think you want to take a stab at it.
Ooh, we'll give a couple minutes for that.
I don't see any.
Nothing yet.
Nothing yet. It's okay. You don't have to answer. So they are mutualism, where both organisms are benefiting. So you can see the honeybee is getting some pollen from the flower. So the bee is getting food, and then the flower is getting a benefit by being able to propagate by the pollen being spread. And then the other kind is commensalism. And this is where one organism benefits and the other has no net positive or negative associations with this. So clownfish and anemones. The clownfish get protection from the anemone, and they get a home and protection from predators. And the anemone is not really bothered by it. Nothing happens that's good or bad. So those are just two examples, and I'm sure you can think of lots more, but it's something to think about as we go through this. And sometimes symbiotic relationships are not always clear. So this is an example of a Greenland shark, which are very cool. And Greenland sharks often have these parasitic copepods, and these are attached to the cornea and they cause blindness. So you can see that the eye of the Greenland shark is kind of fuzzy and you can't really see defined features. So it's blind. And obviously you can say, oh, wow, this has got to be really harmful for the shark, right? The shark can't see. Obviously, that's a parasite. But the parasitic copepod can actually serve as a kind of a lure and attract fish and other things that the shark likes to eat. So it can actually benefit the shark. So sometimes it's not always clear whether a relationship between two organisms is mutualism, commensalism, or parasitism. So it keeps kind of us on our toes and keeps us wondering and thinking about things a bit more deeply and more about how there could be advantages and disadvantages to a lot of these different lifestyles. So we have our first poll here. I want you to look at these 8 pictures here and these 8 organisms, and I want you to guess how many of these organisms are parasites. And I think there's a few different options that pop up. So do you think 2 of them, 4 of them, 5, or all of them are parasites?
Ooh. Getting a mixed range at the moment. So we'll just
Okay.
We've got someone in the chat box who thinks all.
Okay.
I'll just give a couple more minutes because we've got half the responses. Here we go.
Yep.
See which one wins. These are some lovely photos, though.
I know, aren't they beautiful? None of them are mine, unfortunately.
Oh. And number 5, is that one we're going to see hopefully soon with the season?
Number 5, sorry, can't see with. Oh, yes, exactly. Yep.
And share the results. So we've got it split between 5
and 8.
Yeah.
You guys are very clever. The answer is all of them. So all 8 are considered parasites. So next, I want you to do a bonus round because I know you guys are so clever from your answers. Can you name any of them? Do you know what any of these are? You don't have to do all of them. Just pick your favorite one and tell me what you think it is. And it could be a general name or, something specific if you know the specific name.
So we've got tick and mistletoe so far.
Okay, yep.
Cuckoo, number 8.
Yep. Mm-hmm.
Oh, well done. Rafflesia for number 3.
Rafflesia, yeah. I'm always surprised. Someone, it's always the one that I think I'm going to catch everyone on, and someone always knows it.
Very good. We've also got a few others. A hookworm, a tick and a tapeworm, and bloodworm. Are those some of the options?
Not the bloodworm, but yeah. Mm-hmm. Well, yeah.
A virus, tapeworm, stinking corpse lily, cuckoo, tick, flower from Super Mario. Yeah.
Yeah.
Looks like it.
Yeah. Yeah.
We've got COVID. So do you want to give us the answers?
Yeah, that's awesome. You guys did really well. So we have tapeworm, a nematode, Rafflesia, which is a parasitic plant, viruses, mistletoes. Mistletoes, a lot of people don't know, but mistletoe is a parasitic plant because it attaches to a host tree or shrub and extracts all the water and nutrients from the host plant. So that one's to keep a lookout for that one. I think it might actually grow here. I'm not sure though. And then we have the tick and bacteria and the cuckoo. And the cuckoo is really cool because you can see the cuckoo, the baby is the big bird on the bottom there. And mom is, the surrogate mom is the top one. So cuckoos lay their eggs in other birds' nests and then leave them there. And then because those birds are usually much bigger, they out-survive and compete the other, native birds' babies, and they get all the food from the mom that's not actually their mom. So they're pretty sneaky.
But smart.
Yeah. So lots of examples of parasites here and how different they can be and how you can find them all over the place. So today I'm going to be talking to you about parasitic worms, and we call parasitic worms helminths. But helminths is a very general term, and it's not really a taxonomic word that is, a formal name for any group. It's very informal, and it includes both nematodes and platyhelminth species, which are not closely related to each other. But when we talk about helminths, we're talking about both of these groups. And I put this in there because I always think it's really interesting. Everyone is really grossed out by parasitic worms, but a lot of people actually deliberately eat parasitic worms. They can be for perceived weight loss benefits, but it's actually very unhealthy. To treat disease, like a lot of people can use helminths to treat allergies and all sorts of autoimmune disorders. You can shock people with it, like this guy in here, he's being a bit cheeky and gain social status. People do it for scientific purposes. If they want to study a parasite, you can see what it does in yourself. They eat them while they're grooming. And it can also be a coveted source of nutrition. This is a pretty famous French dish that uses a roasted woodcock, and the intestines are infested with a little tapeworm. And it's said that the taste and the delicacy and the flavor of this dish is due to this tapeworm. So it's actually a lot more common than we think. And now we have now we have our second poll. I want you to guess how many people in the world are infected with at least one helminth.
So that's being nematodes and...
Yes, all of them. Nematodes, trematodes, cestodes, all the parasitic wormy things. But only people. We would never be able to know how many animals are actually infected because there's just too many.
It's interesting in the chat box. We've also got someone with the emoji of a vomiting face. I was feeling a bit, oh, no, I don't want to try that one. No, I'll take this one first. Oh, brilliant. Response either in the chat box, either C or everyone, and I'm just going to end the poll and share the results. So majority say 2 billion.
Everyone wasn't an answer.
Everyone has
2 billion. You guys are awesome. I should have made these questions harder. That's really good. Yes, 2 billion. I thought I'm tricking everyone, but you guys are too smart. Helminths are literally everywhere. They infect almost 2 billion people, and we don't even know how many animals are infected, like we said, but almost every animal has had a helminth infection, and almost every human has had one at some point in their life. And also co-infections are really common, and co-infections are when you have more than one worm at a time. They can be found in every habitat. You find them in deep sea vents all the way to the top of the, tallest mountains. Everything is infected everywhere. And they can be really, really tiny, less than a millimeter long to several meters. So the one on the right is one that I collected when I was much younger during my PhD studies. And this came from a cow at a slaughterhouse that we were at, an abattoir. And this is only a piece of it. So you can see they can be very, very long and huge and probably can make animals and people very sick.
Is there any limit to how long they can be?
Yeah, well, so it usually, so it depends on the species, of course, but also it depends on the size of the host. So usually when you have a larger host, you see a lot bigger parasites. Because the parasite wants to keep you alive too. They need to keep using you to feed off of and use your resources. So they have to, minimize the amount of damage that you do. So they don't want to grow to be really huge and, bursting out of you so that you end up, dying really quickly. They have to kind of moderate how bad they are. But you can become infected through a lot of different ways, depending on what the worm is. It can be through eating it. It can go through your skin in some cases. It can be transmitted by insects. So there's a lot of different ways that you can become infected. And I bet you that even you have had a parasite before, which is something you're going to have to go ask your parents about if you don't know. But pinworms, or they're called threadworms commonly too, are very common in nursery and school-aged kids. So I bet you've all been to school. Most of you have been to nursery. You've probably had a threadworm before, and you can pick up tablets to get rid of them from boots. So they're really common, and you can go home and talk to your parents about it, and they'll probably remember, oh my gosh, yes, it was so disgusting.
Probably. We've got someone who did say that I do, when you were talking about it, everyone's got one. And then we've got a question about, how does infection by skin penetration work? How do they get through your skin?
So one of the parasites that I'll talk to you more about later, it goes through snails. So it has an aquatic stage, and parasites often have really complex life cycles. They don't just develop in you and leave and then go into another person necessarily. Sometimes they go through mosquitoes, snails, other organisms first. So the one that I'm going to talk to you later about, it's found in freshwater snails, and it goes through developmental stages within the snail, and then it comes out of the snail. It escapes out of the snail skin, and then it kind of follows the water current and also can move itself to find a human skin. And then it will use enzymes that digest your skin to get through your skin. And all you feel is an itch. So your skin will start itching, and you don't really know what's happening. But there can be worms later on.
Does that process take a while? Or someone's asking, how long does it take to penetrate the skin?
So it can take up to 24 hours to get through the skin and to find a blood vessel. So they go and they find a blood vessel, and then they travel through your vasculature until they end up in the veins of your intestines. So that particular one lives and eats your blood. So
fantastic.
Yeah. Yep.
Thank you.
So how do we control helminth infections? There's several different ways, and usually involves more than one of these. Healthcare intervention is one of them. Improved infrastructure. So a lot of worms could be eliminated if we had better sanitation, better access to clean water. We can educate people about how they become infected so they can learn how to avoid infection or learn the symptoms so that they can get treated quickly. And then what I do is doing research on them, which may not see, immediate impacts from doing research. It's more of a long-term game, but in complement to these other factors, is really working to eliminate a lot of these debilitating diseases. And genomics comes into play here because it's a very powerful tool that we can use to understand these worms. And we do that by looking at the genome, the transcriptome, and the proteome. So when people talk about genomics now, they're not just talking about the genome, they're talking about all these other components of the genetic makeup of these organisms. So next we have our poll, and I want you to guess which molecules are shown left to right, and I purposefully took off the labels that just had them on there. So tell me what you think is being shown in these.
So while everyone's filling out the poll, we've also do the worms penetrate the brain once inside the host, or do they stay in your intestines?
Okay, that's a good question. So sometimes this parasite that I was talking about, there's a lot of different species that can infect humans. Some of them infect mice, some of them infect cows, and some of them infect birds even. And sometimes if they go into the wrong host, you can have them found in places they're not supposed to be found. So there is actually one bird parasite, a bird blood worm, that normally infects birds, but if it infects humans, it can go into your brain. And it's very, very rare and doesn't happen very often, but it is possible. And that's true with a lot of worms, is they can kind of get lost and go different places, and they can cause a lot of problems.
And then we've got another follow-on from that. If the parasite ends up in sputum, does it mean it's reached the brain?
No. So, no. So any parasite in the brain will typically cause like encephalitis type symptoms. I don't know about it being found in the sputum and having that being evidence. I think that would be evidence that it would be more in the gut or in the lungs, I guess.
Interesting. So we've got our results. So majority of people say it's DNA, then RNA, then proteins.
Awesome.
Oh, goodness, they're so bright.
I know. I'm telling you, we'll have to do a redo and I have to make harder questions.
Yeah, next time harder.
Yeah.
Very well done, everyone.
Excuse me. So we talked about genomics and how genomics can be, talking about the genome, the transcriptome, the proteome. So the DNA that you have, the RNA, and how that makes genes, and then how these encode for proteins, right? So the thing that I use is called functional genomics, and it's kind of a new, exciting field that is using multiple of these tools and these technologies to uncover how biological processes are actually happening and describing an entire system. So looking at, not just what the genomics of something is, but looking at what the function is and looking at how this produces a specific phenotype or a physical trait. So I'm combining genomics in terms of looking at the genomes and the DNA and transcriptomes to perform functional genomics experiments. And I'll kind of show you how I do that and what that entails. So when I told you about these worms, the worms usually have different sexes. You have male and female worms. Some worms, some parasitic worms have both capabilities, so they can produce asexually, but most of the parasitic worms have male and female worms when they're adults. And this is the worm I was talking about before that can go through your skin in its infectious stage, and it's called a schistosome. And it's called a parasitic flatworm. And put your hand, I think you can do hands up, right? Put your hand up if you've ever heard of the free-living flatworm, the planarian. So you might have had science classes where you chop these up and they regenerate. They're pretty famous for doing fun experiments on regeneration. Has anyone ever seen them, heard of them, or even done any fun experiments on them?
We've got our 6 hands up so far. 7.
Oh, awesome. That's cool.
8. Oh.
Wow.
I never did this at school.
No, it's really fun.
Oh my goodness. Someone just put in the chat box. They are so
They are
cute.
very cute. Yeah, they're very cute.
Sure.
You can chop them up into a million little pieces and they'll regenerate into new ones.
And that's why they're cute. Yeah.
So the parasitic flatworm that I've studied the most is called the schistosome on the right. And it causes a disease called schistosomiasis or bilharzia, and it's a neglected tropical disease, which means that it's very prevalent and it's neglected in that there isn't enough research on it, and a lot of people get very sick from it, and more work needs to be done towards eliminating it. It can cause really serious, debilitating clinical symptoms. It can result in death. And it mostly infects women and children. So It can cause a lot of problems in development of children and developmental delays physically and mentally. And so here's the worm in a picture. You can see the male and the female. So the male is the pink-looking one, and the female is the really skinny, green-looking worm within it. So I like to describe it kind of like a hot dog and a hot dog bun. So the male is like the hot dog bun, and he holds the female in this groove that he has, and the female just sits in there and eats blood from its host.
Sounds like a delicious
Yeah. So there are estimated 200 million people that are affected, and greater than 90% of these cases occur in sub-Saharan Africa. And so the world's poorest and most vulnerable people are affected by this. And the bad thing is there's only one drug that can actually treat it. And this drug only kills the adult worms. So it only kills them after they've already developed. So even if you take the drug, you might still have some worms that are not completely developed yet and are resistant to the drug, or you might get reinfected again, which is very common. So it's not really effective in eliminating the parasite and is what is causing it to be so prevalent. So how can we stop the worms from producing eggs? This is the big question that I have. So parasitic worms produce a lot of eggs, hundreds of thousands a day. And so all of these eggs are what get caught in the veins, get caught in the tissues, and these are what are causing most of the symptoms because the body has really large inflammatory responses to these eggs, and they can get stuck in places, and they can cut off blood supply, cut off all sort of fluids from flowing in the tissues. So what I want to know is how can we stop worms from making eggs in the first place? And they only make eggs because they have males and female worms. So how can we stop males and females from developing altogether? So what we know is that most helminth species have separate sexes, but we don't really know how sex is determined. We only know that sex is determined genetically when the egg is fertilized. So it's encoded in their DNA, just like us. So when we are formed in our eggs, when the egg is fertilized, immediately you're either male or female sex. So we know that this is how it works with worms, but we don't know how the mechanism works and how we can manipulate that to control them. So this is an adult worm pair. So the male is the chunky one and the female is kind of poking out. And they have sex chromosomes like we have sex chromosomes, except theirs are called Z and W. And kind of opposite to us, how we have males have X and Y and females have XX. It's the opposite with these worms where males have ZZ and females have Z and W. And so we want to know how we can manipulate this and how can we uncover what is driving males to become male and female to become female. And the first thing we look at is the genome. And so it all starts with having a complete and annotated sequenced genome. And what do we mean when we talk about an annotated genome? So first we have our sequenced genome, including the sex chromosomes. And this was an effort that took about 10 years. So it was a really long process. And just to give you some example of how difficult this was, there are only 5 species in the entire world that have their sex chromosomes sequenced. And one of them is the human. So this is really rare to have sex chromosomes sequenced, but it's the key to understanding how male worms become male and female worms become female. And then obviously to manipulate them after that. And then annotating the genome means to accurately understand and identify where all the genes are in the genome on every single chromosome. And so all the gene changes and new genes that were described in this latest version of the genome. And this took significant effort as well. So the understand we have the genome is I wanted to look at the transcriptome. And so the transcriptome is what actually is being expressed of the genes. And this looks really scary, but I'm going to go through it with you and tell you all about it. So heat maps are a visual tool that we use to look at different trends in gene expression from sequencing the mRNA, which becomes the genes. So on the left, we have our gene names, and these are just randomly generated names from a system. And then we have our sample names on the x-axis. And then this color coding that you see for each of these genes is used to tell us how much a gene is being transcribed. So how many copies of them are there? So if you see a yellow or a green color, that means that there's high gene expression and that gene is really important for that sample at that stage. And if you see a dark color, a dark blue, that means there's no expression and that gene is not transcribed. So this is telling us how or what genes are important in males and what genes are important in females while the worms are developing and becoming adult worms. So they can tell us or give us hints into what genes are used to make them into reproducing males and females. So first we started out with our genome where we sequenced and annotated the sex chromosomes. And then we found out what genes are expressed in males and females. And now we want to get to what the functional genomics part is. And so one of the experiments I've done that's considered functional genomics is looking at single molecules of transcripts. So single transcripts that are found, and these are expressed in different parts of what is shown as the head of the worm. And you can see where the arrows are pointing is each little dot of color is showing a specific gene that I was testing. So I found a gene that was only expressed in females, and that's the pink one. And I wanted to see where in the worm is this being expressed and how it could be important to the development of the worm. So what we found was this gene that's pink here is also being expressed in cells that are important for or known to be developing in the worm gut. So there's something specific going on for the worm with the development of a female specific gut development that we're looking at here. And you can see an increased magnification in the right side up to 63x. And each of those tiny little dots is one transcript that's being made in that cell. And the white part around it is the nucleus. So we can also see where in the cell these transcripts are and if they're exported out of the nucleus or not. So this is one of the really cool, and I think it's really pretty. I like all the different colors of it.
With you on that.
Yeah. So it's really complex, but it's just giving you an idea of some of the experiments we can do to uncover how the genome actually translates into a meaningful biological result for us. And then also, this is less of the hardcore science, but I wanted to show you guys what science really looks like in the lab. It's really fun. Those images are years and years of work, but on the day-to-day, we spend a lot of time working with worms in a sterile hood. We spend a lot of time on microscopes and pipetting things, and there's a lot of experiments that need to be repeating. And so it's a lot of fun. Everyone has a really great time. And my favorite part is, as a parasitologist, this is the best thing about studying worms, is you get to go all over the world. So I've studied on, I don't know how many different continents. I've done a lot of field work all over the place. I've met amazing people. I've got to train people in worms and how to study them in their own countries. And I've gotten to see amazing things along the way. And now you're probably wondering, being a parasitologist sounds pretty cool. So how do I get to be one? And you can go whichever. I became a scientist by studying microbiology. So I studied bacteria first. I was born and raised in Michigan, and then I decided to go work for a few years after I got my undergraduate degree in science, and I wasn't really sure what I wanted to do next, so I took some time off. I traveled, I had fun, I worked in a lot of different labs and kind of got a feel for what I enjoyed doing. And then when I was a bit older, I went and did my PhD at the University of New Mexico, and that's where I started studying parasites. So I didn't always study parasites. I didn't always know what I wanted to do. I knew I wanted to be in science, but I didn't know what, and I didn't know how. I didn't know how to get there. And I didn't particularly do very well at uni. So there's always hope. I had a bit too much fun. And that caused a little delay in my career, but it was totally worth it. And I learned a lot along the way. And then after my PhD, I moved to the UK for the first time. And then during my PhD, I did a bunch of field work in Africa and the Caribbean and all over the States. And then after I went to the UK, I went back to the States for a year and decided, no, that was a bad idea. Get me out of here. And now I'm back in the UK. So, being a scientist isn't always this straight path. You can do a lot of different things and go and discover, different areas of science that you enjoy, or not do science at all for a while. But, it's all fun in the end, and you get to explore and do a lot of cool things. So now, if you have any questions, I will take them. And this is a dog hookworm. Sorry.
How many magnification is that?
Oh, I don't know. Probably, yeah, I have no idea.
It's quite veiny as well. Yeah. Lovely.
Yeah.
Well, thank you so much for that, Sarah.
Yeah, no problem.
Questions are ready for you.
Okay.
So just back on one of the slides that you had that had a couple pictures of especially a person with a swollen abdomen.
Yeah.
We had a question of that. Is that what we see famine sufferers, the swollen belly?
So,
yeah.
Sorry, I just went past it. Yeah, that one. Yeah. So, I mean, addition and yes, when there's a famine, you have a distended belly, but it's also a symptom of a lot of different things. And with schistosome infections, the egg buildup in the veins causes the veins in your intestines to become distended, and also your liver and sometimes your bladder, depending on the species. So it's a symptom of a lot of different things. But in this case, this particular person has a very heavy schistosome infection.
That must be painful as well.
Yeah.
And then we've got another question about, do parasitic worms have any toxins that they release on entry, so when they enter your body?
No. So they're very good at evading the immune system. So they have this outer covering on them, and it is continually replaced. And so they can kind of hide themselves from your body's immune system. And other than secreting the enzymes that, dissolve your skin, which you don't really feel, it just feels like itching. No, they just go straight to your bloodstream, get in there and start their path.
Lovely. So very good at being ninjas there.
Yeah.
Oh, very nice. And then we've got another one. What has been your most interesting study and why?
Oh, so one of the things I did during my PhD that I really liked was there was a study. It was more of an epidemiological study. So I think the coolest thing about being a parasitologist is that I never do the same thing. I'm always doing something different. Like, there's mathematics involved in modeling disease. There's computational work. There's field work. There's, general biology work. You do a bunch of different things, which I really enjoy. I like doing a lot of different fields all in one. But what we did was, I think it was, oh, yes. So this right here, can you see my pointer? So this is a stream where there's heavy infection in Kenya, and we wanted to know how people are getting infected and where and how much. But obviously you can't do these experiments in people. So what we did was create these floating cages of mice, and they had mesh on the bottom, so just their little feet could go into the water. And then we put them every 30 meters down the stream, and we wanted to see a few different things, like would the mice become infected with the worms? How many worms did each of them get? And then also, what was the genetic diversity of them? So we could sequence the worms and figure out, how closely related they are. And it's really interesting because the worms reproduce asexually in the snail host. All of them are genetically identical. So you can have a mouse that's infected with the same genetically identical worm several hundred meters apart, meaning that one snail infected with one parasite can infect many different people downstream. And you can also find what we call hotspots of infection, where people will get infected really readily. And this has direct applications. And that's one of the reasons I like it, because you can tell people, hey, this spot in the stream, don't go in there. You're going to get infected. Stay away from here. Try going over to this spot instead. But I thought that was a really cool experiment. And unfortunately, it was never published. And that kind of happens sometimes. Not everything is always published, even if it's the coolest work that you do. But I got to tell a lot of people about it at conferences, so it's still out there.
Still out there. That's good. Very interesting. We've got quite a few more questions. So we've got which parasites are visible to the naked eye?
Mm-hmm.
There's quite a few, isn't it?
Yeah, I mean, in terms of worms, all of them are visible to the naked eye when they're in their adult stage. There are certain stages of the life cycles that you can't necessarily see with the naked eye unless there's maybe a whole bunch of them. But all the worms, all of the worms you can see.
And then we've got, I'm going to attempt to say this word. Have you worked on
Strongyloides? I see it. No,
oh,
I have
goodness.
not. I have not, no.
Oh, lovely. Is there any programs that do clinical testing for people with diagnosed parasitic infections?
Yeah. So the World Health Organization has set up stations all over the world for testing. Most of the time, the testing takes place at schools. So this is that school picture of those kids on the left. That was one of the schools that we've gone to. So we collect fecal samples from them to collect worm eggs, and do a bunch of experiments that way. But we also take a doctor with us, and the doctor will treat any infections that we confirm that they have. So, and this is done really prominently all over the world. The only problem is that some of these rural areas are a bit harder to get to, and they may not get treatment as much as they need it.
Unfortunately, always the case with diseases in general as well, isn't it?
Mm-hmm, mm-hmm.
We've got, so another question, human bodies can reject transplanted organs since it's foreign tissue. So why aren't parasitic tissue rejected and destroyed by our bodies?
Yeah, so this kind of goes back to what I was saying before, where worms are really good at regenerating the outsides of their bodies. So, once they detect that the host can see them, or the immune system can see them and starts going at them, they can regenerate the outside so that they're invisible again, essentially. So they produce they present different antigens on the outsides of them so that they can't be detected. And they continually do this, and it's both to evade the host immune system, but they also do it naturally as they grow and get bigger through the life cycle. So they continuously shed and keep getting bigger and also evade. Why do corticosteroids cause hyperinfection? I'm not sure what. And strongyloides.
Oh, but you haven't worked with those ones.
Oh, okay. So, why do corticosteroids cause? I don't remember why. I'd have to look that up. I'm not really sure.
Fine, we can add that to a question for a later date.
Yeah.
Yeah, cool. So that seems to be the most of the questions for now. Do you mind stop sharing, Sarah?
Yep.
Awesome, fantastic. Thank you again so much for that. I'm just going to make another poll live and relaunch it because you've talked a lot about genomics today. So just wondering how everyone's knowledge of genomics has improved. We had a bit of a mixed batch earlier, so it'd be nice to see how Sarah's talk has influenced you. I found that so fantastic and a bit squeamish. I worked with malaria parasite before, and I think worms, a whole different ball game there.
Yeah.
Yeah. Do you know what's next for you in your career?
Yeah, so I studied schistosomes for about 12 years, and now I'm switching to work on nematodes. So a bit different, but the same idea. How are they developing into different sexes, and how can we use this? So I'm focusing on using a sheep nematode as my model organism, and doing a lot of experiments and cool different things with that.
So will you get those specimens? Will you get like sheep poo and stuff like that from around?
Yeah, we have sheep that we have in Scotland. We have a flock of sheep that are infected regularly, and we get all sorts of parasite stages. And yeah, I think the picture that was the ad for the talk, those are nematode parasites. Those are the parasites I study now.
Very nice.
Yeah.
Someone's
It's
saying fasciola?
not fasciola. So no, fasciola is not a nematode. It's a liver fluke. So that's more closely related to the schistosomes. So the nematode I'm studying is called Haemonchus contortus, and it's found worldwide in small ruminants like sheep and goats.
That's quite a name as well. Can you spell it?
Yeah.
I'm putting that in the chat box. Thank you. Although a lot of these words don't ask me how to say. But thank you again so much. So we've got still a couple minutes, but I'll just say that our next genomics session will be on the bacterial population on the 26th of January. So hopefully everyone will have a very lovely Christmas break. Do you have exciting plans for Christmas, Sarah?
Working, catching up on work.
Oh, don't work too hard. It will be.
No, I know. Yeah. Well, it's too expensive to go visit family, unfortunately. So we'll stick around and have a very British Christmas. And yeah.
British Christmases are fun. Maybe it'll snow. It is getting very cold.
Yeah, it is. It is. That would be nice if it snowed.
Yeah. I bet you're used to the snow in the States then as well.
Yeah. Yep. Mm-hmm. Yeah. It's a lot colder there. A lot more snow. Yep.
Yeah, I think someone told me it's a few meters. So we've also got from the chat, very interesting. And Merry Christmas and Blessed New Year's. Thank you so much.
Thank you guys. Thank you so much for joining. I'm really hoping you enjoyed it, and don't think parasitic worms are so awful anymore. They're pretty cool.
No, they are awesome, but I don't know if I'll try that cuisine anytime soon.
Well, you've got to try everything at once, I think.
Have you tried it yourself?
I would. I have not. Maybe I can make it for Christmas. It looks like a Christmas. It looks like a Christmas dish.
Grab some from the lab and just no. Yeah, very interesting. And also, so we've had just a question about if they have any more questions for you. So if anyone does have any more questions for Sarah, they can email us at engage@welcomeconnectingscience.org, which I'm just going to put in the chat box now.
Yeah, I'd love any other questions. I love talking about parasites all the time. And Christmas is a very productive time. Yes, it seems like everyone typically somehow miraculously stops working about 2 or 3 weeks before Christmas. And so that means I have the entire lab to myself and I can play my music and do everything I want to do.
Yeah, we've got a good campus closure time at that time. It's like a ghost town. So do intestinal parasites eat each other?
No, but in snails, there's different species that will eat each other of the parasite within the snail.
The poor snail.
I know, the snails really get the raw deal of it, to be honest.
Yeah, the poor things. Oh, yeah. Well, we'll finish a couple minutes early then so everyone can have the rest of the evening. Thank you again so much, Sarah. This has been so fantastic.
Yeah, thank you so much, everyone.
Oh, lots of applause, lots of applause. Love it.
Good.
Thank you. Have a good evening.
You too.
Take
Bye.
care. Bye.